It's my pleasure to introduce Dr. Laura Lewis today. So Dr. Lewis is faculty at Boston University, and she got her PhD from MIT, where I was lucky to get to know her and to befriend her. After getting her PhD, she postdoced at the Martino Center at Mass General Hospital as a junior fellow at the Harvard Society of Fellows. And she's currently an assistant professor in the biomedical engineering department at BU, Boston University. So Laura has received numerous awards for her groundbreaking work, from the One Mind Rising Star Award, the Sloan Fellowship, the McKnight Scholar Award, the Pew Biomedical Scholar Award, and many, many others. And she gets these awards because her research is super cool and innovative, right? She pushes the limits of what we can achieve with neuroimaging. And she uses multimodal methods, including fMRI and EEG, and she studies sleep and arousal. And today, I hope that she's going to be convincing all of us to use new methods of fast fMRI. So please join me in welcoming Dr. Lewis. Oh, I think you're muted. Laura, yeah, there you go.
Can you hear me? Great. Thank you so much for that incredibly generous introduction. I'm super excited to get a chance to talk with all of you today and present some of our work on imaging of sleep. And yes, as Zainab has mentioned, I am an advocate for these kind of new fMRI methods, so I'd love to talk about those as well. So I'll just be discussing some of the recent work from our lab that's focused on understanding the neural and also the physiological changes in the brain that happen to us during sleep. So sleep changes almost every aspect of brain physiology. And so the one that we're really subjectively aware of every night is perhaps the cognitive effects of sleep. So we shift from being very engaged with our environment to states of unconsciousness or dreaming. I think we've all also experienced the cognitive effects of not sleeping. So even just one night of bad sleep can really affect your mood, your cognition, and your attention the next day. These kinds of diverse cognitive consequences of sleep are probably related to the fact that sleep seems to change neuronal activity in essentially every part of the brain. And so sleep dynamics are typically measured with EEG in humans. And you can see from the EEG of somebody transitioning to non-rapid eye movement sleep, there's this really dramatic change between this very low amplitude desynchronized EEG during wakefulness to the appearance of individual oscillations in the EEG during sort of mid-sleep with these spindles and individual slow waves. And by the time people transition into the deepest stages of sleep, the EEG is showing these really large slow waves. This is reflecting that neurons really throughout the cortex and most of subcortex are really changing their firing patterns across these different sleep stages. And these patterns have been used now for decades to classify what type of sleep stage somebody is in. In addition to these kinds of cognitive and neuronal consequences of sleep, more recently it's been realized that sleep also seems to play a special role in waste clearance from the brain. So while we're awake, neurons are kind of metabolically active. They're generating waste products, including things like amyloid and tau. And during sleep, the levels of those proteins in the brain also seem to decrease. So it seems there's also these kind of lower level metabolic consequences of sleep for the brain. And so the core question that our lab is interested in is, what is sleep doing in the brain and how is this regulated? So this cartoon shows just a handful of the deep brain structures that have been shown in animals to be able to actually causally control sleep and wake. And what's sort of striking from this is that these neuromodulatory centers are essentially projecting throughout the entire cortex and the entire thalamus, really reflecting that it's maybe no surprise that activity throughout these really distributed networks is all changing across sleep and wakefulness. And so the switch between arousal states really reflects a modulation of kind of whole brain level circuits and whole brain physiology. And so that means that to understand what's regulating sleep and what its consequences are for the brain, we have kind of a technical challenge in terms of our imaging. We'd like to be able to measure many different things at the same time. The first is these kinds of fast timescale dynamics in the cortex, especially these kinds of EEG rhythms that we use to determine if somebody's asleep, but also really kind of measuring throughout the cortex to understand how large scale networks are affected. We'd also really like to be able to image the thalamus, which we know is a kind of core region for coordinating rhythms throughout these cortical networks, as well as these kinds of deep brain neuromodulatory nuclei that seem to be important for controlling sleep and wakefulness. So ultimately, we want to have this kind of fast oscillatory measures in the cortex. We want to be able to measure deep in the brain at relatively fast timescales as well. And we'd like to understand not just what the neurons are doing, but potentially also what non-neuronal changes are happening in the brain during sleep. And so the approach we've been taking is to try to really integrate multiple different imaging methodologies to be able to look at this in humans. And most of these methods are using EEG, which lets us really kind of assess the sort of millisecond scale dynamics of the brain in concert with fast fMRI, ultra high field fMRI to look at tiny deep brain structures, as well as different contrast mechanism in fMRI to look not just at the bold signal, but also at other elements of brain function. And so just to give a quick outline of what I'm planning to talk about today, I'll first introduce fast fMRI. I think most of the audience here, this is the fMRI community, so you might be interested just in kind of the technical developments along the fast side. And then using this method to look at two specific questions on sleep. First, what are the neural circuits that engage at the moment that we transition between states and seem to reflect this kind of switching property between these really different operating modes for the brain? And then second, how does the widespread change in neuronal activity that we see during sleep relate to these other consequences of sleep, potentially things like the idea that waste clearance is changing in the brain during sleep? And to do this, we'll use multimodal neuroimaging to try to kind of simultaneously measure multiple different things about brain function during sleep. So focusing on the first one, this idea that fast fMRI can potentially serve as a new tool for measuring neurophysiology in humans. So about 10 years ago now, there was a significant advance in the temporal resolution of fMRI. Conventionally, acquiring a whole brain image took 2 or 3 seconds as we kind of slowly stepped through each individual slice of our 3D volume. And with the development of multiband or simultaneous multi-slice acquisitions, it's now possible to get many slices at a time. And so if you acquire 8 slices at a time, you can image 8 times faster. And so it's now possible to do whole brain or near whole brain acquisition in timescales of, for example, 300 milliseconds or so. So acquisition is no longer the constraint. We can actually image quite rapidly. And so the key constraint is really what is the biology that we're imaging. And so as you all know, when we're doing fMRI, we're measuring not the neuronal activity itself, but rather the kind of response of blood flow and blood oxygenation that takes place after neuronal activity. And that's typically been assumed to follow this kind of slow time course, with hemodynamic responses peaking maybe 5 to 6 seconds after neuronal activity and really unfolding on a slow basis. And so because of this, these models would essentially predict that you can scan as fast as you like, but the signal that you're measuring is slow. And so there may not be, from that perspective, much of a benefit to scanning faster if you're actually studying something that's slow in the first place. And the key thing to recognize is that these models of the hemodynamic response really come from sort of foundational work, 20 to 30 years ago, where people were using very slow stimuli in long block designs in primary sensory cortices. And it's already well known that bold responses are actually nonlinear. The properties of these responses changes depending on your stimulus, depending on your brain state. And so we felt it was actually an open question, how fast could this potentially be? And to measure this, we decided to actually go in and ask, if you scan faster, what is the ultimate temporal resolution that you could achieve with a bold signal? And to test this, what we thought is basically if we could induce the neurons to fire at an oscillation that we know and we control, we can then go in and measure with fMRI whether we can actually track that oscillation in the neurons as well and test ultimately how fast are we able to go. Conventional models of the hemodynamic response predict that you shouldn't really be able to go above 0.2 hertz or so kind of an oscillation period of 5 seconds. But if you actually look at this paper from 2000 from Benettini and Cox, they already were showing this 2 second on, 2 second off paradigm. They were starting to see hints of a bold response, which is already at the 0.25 hertz level, which is quite a bit faster than these conventional models predict. So we decided to kind of follow up on this and design a visual stimulus that would let us change firing rates in the cortex in a way that we know and control, since we know that we can get really robust responses in the visual cortex by having really high contrast flickering visual stimuli. And so the way this is designed is that the luminance contrast of our stimulus would kind of oscillate at some frequency that we control. So this is what it would look like. It gets brighter and darker. So firing rates will go up and down and up and down. And so then by changing the frequency of this luminance oscillation, we could change the frequency at which firing rates within primary visual cortex should be changing and go ask how quickly can fMRI keep up. And so I'll just kind of go very briefly over what we found. We started with what was thought to be kind of the upper limit of fMRI, this 0.2 hertz timescale. And we actually found that if our stimulus is cycling over 5 seconds at 0.2 hertz, this is the mean response in primary visual cortex. And you actually get a very robust oscillation in the visual cortex, even at this frequency that was supposedly at the kind of upper edge of detection of fMRI. We then tried to push the frequency and went up to 0.75 hertz, where now we're seeing dynamics unfolding on timescales of hundreds of milliseconds, as the full period of the cycle is about 1.3 seconds. And note that the y-axis of this is 100 times smaller. So your signals do become tiny, but we could still very reliably elicit this oscillation in the fMRI signal in the visual cortex. So it turns out that actually you can capture hundreds of millisecond timescale dynamics with fast fMRI. And while these signals are small, they're actually an order of magnitude larger than would have been predicted by conventional hemodynamic models. And what I'm showing you is best case scenario, because the visual cortex is big, it has huge bold signals. This is kind of the easiest thing to do. And of course, what we like about fMRI is that we can image the whole brain, not just the visual cortex. So we did a follow-up study asking, could we use this for a brain structure that's uniquely accessible by fMRI, which is the thalamus. So this kind of red functional localizer is popping out here, the lateral geniculate nucleus, which is the primary visual thalamus. And this is a much more challenging region to image because it's very tiny, it's deep in the brain, far from the coil. And we repeated the same study, going just up to 0.5 hertz this time. And again, we replicated that we can see these cortical oscillations in blue, but we also found we could really robustly measure these visual evoked oscillations in the LGN, the visual thalamus as well. It's a little bit noisier, which makes sense because it's this much tinier structure. But in fact, this technique seems to also be effective in these small, deep brain structures. And one thing to just note is the thalamus is early here, and this is a really long time delay. So this is probably not a neural time delay, but instead is reflecting what a few studies have recently found, which is that hemodynamic responses are faster in the thalamus. So this oscillation will actually appear earlier in time. So this is kind of the technological conclusion, which is that
Laura, can I ask a quick question?
Yeah.
Can you go back one slide?
Yeah.
So I'm wondering, is there any meaning to the fact, like in the slower oscillation, the signal starts off and sort of ramps up at the on signal, but for both of the faster ones you showed, it seemed to sort of start and then dip down. And so what's the interpretation of that? Is that meaningful?
That's a great and important question. So the key thing to note is that even though timescales are fast in the sense of replicating temporal precision, there's still a 5 to 6 second lag. So when you plot something with a 2-second cycle, you get phase wrap. So essentially, you still have your 5-second lag with your signal. So what the peak time is kind of displaced by the phase wrap of that oscillation, if that makes sense. So for example, here, a peak at 1 second after your stimulus, that's unlikely to be the case. It's probably actually 6 seconds, right? That's the hemodynamic lag. And the peak at sort of 1.2 seconds here, again, we're probably having phase wrap, and this is really happening about 5.5 seconds after the stimulus onset. And so that's something that, yeah, it's hard to pin down with this kind of oscillatory paradigm where you don't have that specific onset information. Okay, great question. Yes, so fMRI can detect neural activity dynamics of up to at least 0.75 hertz. We don't know yet what the upper limit would be, but we are definitely kind of entering the range where SNR is the problem. So I think it's conceivable you could go to 1 hertz, but these signals are starting to be quite small. But what I think is cool about this is it really kind of brings the useful frequency range of fMRI from this kind of classic 0.1 hertz band that we often look at to look for what we thought were the neural signals, and really expands it up to 0.75 hertz. So we have a kind of broad range where there's potentially useful neural signals, while noting that we have these very strong peaks here at 0.3 and at 1 hertz, which are the respiratory and cardiac signals. So there's of course a lot going on in these fast signals as well. So what I'd like to talk about now is how we've been using these faster imaging techniques to try to look at the Thalamocortical dynamics that are linked to the transitions between brain states. And so we became very interested in awakening from sleep because it can happen very abruptly. As people fall asleep in our scans, it's very gradual. There's a whole spectrum of brain states, and it can be really hard to define a moment that's the transition between wakefulness and sleep. But when people wake up, it's often sudden. There's often kind of this switch where you flip back on and you really kind of re-engage with your environment. And so we were very curious what types of mechanisms might be engaging at this type of transition where you can switch between profound unresponsiveness and wakefulness within just a second or so. And animal studies suggest that one of the core brain regions to examine would be the thalamus. So directly stimulating different areas of the thalamus in animals have in some cases been able to actually cause awakening from sleep or even awakening from anesthesia. And prior fMRI studies have also shown that at arousals from sleep or within the awake state, you'll also typically see a thalamic activation. So it seems that this is a really kind of important structure for potentially governing arousal in the brain. But a key point is that the thalamus isn't actually really one brain structure. It's actually kind of dozens of extremely heterogeneous nuclei that have very distinctive functional and structural connectivity profiles. And so what we wanted to really ask is how this constellation of very distinct nuclei might be changing and awakening from sleep. Is it indeed that the whole thalamus is kind of switching states as we switch between sleep and wakefulness, or are there potentially different functional profiles for the different nuclei within the thalamus? And so this project has been led by Beverly Setzer, who's a really talented neuroscience PhD student in the lab. So this is just an example of kind of what I was talking about. This is from the Allen Brain Atlas. This purple structure is the thalamus, and even just in this one slice, you can see that the thalamus is actually subdivided into a kind of really diverse set of different nuclei that in terms of their anatomy and histology, as well as their kind of functional recordings in animal models, are known to have very different functional roles. And so this cartoon now is showing some of the largest nuclei of the thalamus, things like the LGN, which is the primary visual thalamus, as well as like MD, the medial dorsal thalamus, and pulling out the largest ones that could potentially actually be resolvable with high resolution fMRI. And so what Beverly did was to basically segment out using individual anatomy, the distinct nuclei of the thalamus within each of our participants. And this cross-section is showing, for example, just a few of these nuclei here. So you can see that after excluding any voxel with potential overlap or partial voluming between nuclei, you can see these kind of very distinct regions of, for example, these different thalamic nuclei, which gives us the ability to potentially ask if they're doing the same thing or different things as we awaken from sleep. And so we conducted a study at 7 Tesla to kind of enhance the SNR from these small deep brain nuclei. And the temporal resolution of this study was 247 milliseconds, so that we could start to look at if thalamus is activating during sleep, are there potentially different temporal dynamics across the different nuclei of the thalamus that might help us understand what's happening. And so the way that Beverly did the study was these scans started around midnight. So we have participants who are sleep restricted the night before. They come into the lab at night. They're very ready to fall asleep. And they are just asked to kind of rest in the scanner with their eyes closed and perform a spontaneous task where they press a button on every breath in and out. So we don't have a stimulus that might disrupt sleep. Rather, they're kind of spontaneously self-reporting the behavioral state that they are in. And so what we wanted to look at is what happens at that moment where somebody wakes up, reorients enough to their environment that they're actually able to resume performing a task after at least 30 seconds or longer of a sleep period. And in fact, many times it was, 30 minutes of sleep or an hour of sleep, and then the person would kind of spontaneously awaken and start performing the task again. And this is just a sort of example of what behavior looks like when you do this. So these black dots here are button presses from someone inside the scanner. You can see in this example, we define this moment of awakening where we get our first button press. And this bottom panel here is the EEG spectrogram from this person at the same time inside the scanner. And this is kind of the classic definition of sleep and wakefulness. You see that before they start pressing the button, there's this 14 hertz stripe, which is sleep spindles, suggesting this person's in N2 sleep. And then there's this kind of abrupt brain state shift where these disappear and it's replaced by this 10 hertz stripe. This is the occipital alpha rhythm, which is in general always present when you have your eyes closed, but you're awake. So it seems this behavior is really kind of co-occurring with a switch in the electrophysiological state of the brain. So using this, we can now define these awakening moments in all of our participants and go back and ask what was happening in the thalamus. And so this is the mean signal in the thalamus and in the entire cortex, just averaged together at awakening from sleep, which is the black dashed line. And so consistent with prior fMRI studies, we found that indeed there's this really striking activation in the thalamus when people awaken from sleep and do this arousal transition. But what this red arrow shows is that you can actually see with this kind of increased temporal resolution that this increase in activity in the thalamus is happening at least several seconds before people actually execute the behavioral arousal state transition. So the onset time seems to be about 3 to 4 seconds before people wake up. You get this rise in activity in the thalamus. And then after they awaken, you eventually see this dip back down in both the thalamus and cortex as well. And so this is indicating that we do see thalamic activation at state transitions, but it's actually happening earlier than the behavioral transition itself. So this is, of course, a very coarse representation of what's happening, the whole cortex and the whole thalamus. We also use cortical parcellation to look at all of the different regions of the cortex and ask if all of them are kind of showing just this decline in signal, or whether any of them have this kind of like prior to state transition type of profile. And almost the whole cortex, which you can see here in red, is showing this general profile of declining after people awaken from sleep. But this sort of pop out in red here is showing that both the anterior and posterior cingulate cortex actually seem to be very thalamus-like. They have this activity profile where they have a sharp increase at awakening, and actually, like the thalamus, this actually starts a second or two before the awakening from sleep. So it seems to be a primarily thalamic increase, but also engaging the cingulate cortex at the same time, whereas the rest of the cortex is suppressed at awakening. So there is a kind of distinctive increase in activity in the thalamus at awakening. And then the reason we wanted to do this study was also to start to pull apart what the different nuclei within the thalamus are doing. And so here's just a subset. We can't really plot all of the nuclei on the same plot very easily, but spanning from the centromedian thalamus, which is an intralaminar thalamic nucleus, which is thought to be very engaged in kind of attentional control, to things like the pulvinar, which is a higher order visual thalamus region. And these smooth plots are showing the kind of onset curves of each of these nuclei prior to the arousal at time 0. And what you can see is that although each nucleus seems to activate at awakening, they're really not happening at the same time. There's a very kind of early increase in nuclei such as the centromedian nucleus, which is the intralaminar thalamic nucleus, and only much later onset times in things like the pulvinar and the ventroanteral nucleus of the thalamus. So it seems like the whole thalamus is activating when we wake up, but there seems to be a sequence where a couple of nuclei actually initiate this chain of activity and then others follow. And so this is plotting the time course of just a few of them. But if we looked across each of the nuclei that we were able to resolve with this approach, we actually found this very characteristic temporal sequence locked to awakening, where the CM and VPL are these kinds of early population that activate early, and then they set off this stereotyped kind of chain reaction sequence where we see activity increase across these distinct nuclei, eventually all kind of coalescing into a unified activation, and then the person would actually wake up from sleep. So this was really interesting to us to basically discover that within the thalamus itself, there's a characteristic temporal sequence of activity that precedes awakening from sleep. One really important thing to worry about when you're looking at different timing with fMRI is that because it's a hemodynamic signal, it's possible that just different placement of veins or different vascular anatomy can also influence timing. And so to control for this, we did a second experiment that was just a breath hold, where you can elicit a really widespread vascular reactivity response throughout the whole brain. And this time course shows you the typical kind of bold response you get at the end of a breath hold. You get this sharp rise really throughout kind of all cortical structure, all brain structures, and very consistent with what we already knew. The thalamus leads the cortex a bit. The hemodynamics of the thalamus are actually faster than the cortex itself by about 0.2 seconds. But if we look across the different nuclei of the thalamus, we really don't see a temporal sequence in this case, with the exception of this one AV nucleus. And so it seems that there actually is probably some vascular anatomy, perhaps the position of a large vein in this nucleus that causes it to have particularly slow hemodynamic responses. But this also sort of reassured us that the sequence we were seeing in the past slide with this really kind of distributed propagation pattern across the thalamus could not really be explained by any differences in the vascular anatomy across these nuclei. And then finally, we looked at what happens after people wake up. It turned out some people would just kind of wake up, notice where they were, press a button, and then fall right back asleep again, kind of contentedly sleep for the rest of their scan. Whereas other people would wake up and really remain awake for several more minutes, having kind of fully executed this state transition and reaching a very stable state of wakefulness. And you can see what that behavior looks like here. The purple ones are the people who have these transient arousals where they just wake up for a couple seconds. And then the pink are the people who really wake up and then manage to maintain that state of wakefulness for at least 15 to 20 seconds after the arousal. And so if we went back and looked at the thalamus, what we found is that, in fact, thalamic activity really markedly differs depending on whether somebody stayed awake or had a sort of transient lapse back into sleep. In both cases, we saw this really sharp rise in initial activity, suggesting this seems to really be linked to the transition. But then this later plateau in activity seems to instead reflect whether people kind of sustained a state of wakefulness or fell back asleep. And in contrast, this again is this very kind of coarse measure of the whole cortex. Of course, individual different regions may differ, but the cortex as a whole did not show this kind of global change in signal amplitude that we see in the thalamus across sustained and transient arousals. So overall, we found that the activity of the cortex, question.
Quick question, sorry. So I'm wondering, what do you do about motion with suddenly waking up from sleep?
Yeah, so we have super aggressive exclusion for motion. It's not correctable in my mind when you're trying to look at these very fast timescale differences. So we just threw out anything that crossed, I think, like 0.3 millimeters of motion at an arousal, because it is quite common for people to move when they wake up. And so we decided that just to be safe, we just would not consider those data. And so we only kept the arousals where people actually managed to resume the task without really having much detectable motion in the first place. And I think that that's really important for all of these types of especially sleep-related analyses where motion and your thing of interest can be very correlated.
Laura, I have a quick question too, and I think I must have missed this, but how long was the scan?
Oh, these scans tend to be about 2 hours. It's different for different people because, if somebody's just peacefully sleeping, we kind of let them. If somebody has been awake for an hour and they're clearly like not going to go anywhere, we also let them come out early. We don't keep people in there indefinitely, but typically we allow for about 2 hours. Yeah.
Okay. And then just relatedly, I know you looked at the thalamus, so really interesting results with that and the rest of the cortex, but did you look at maybe other brainstem nuclei like locus coeruleus or something?
So we are so interested to do that, but we don't have the spatial, we traded the spatial resolution here for temporal resolution. And so we don't have the spatial resolution for locus coeruleus in these scans, but we're actually starting new scans now with 1.1 millimeter resolution for exactly that reason. The thalamus is actually, people think of it as this difficult, tiny deep brain structure, but it's actually really big. And these different nuclei some of them are quite substantially sized, whereas something like the locus coeruleus, you really need to be kind of in the millimeter range. And so we're basically starting a new study to ask exactly that question.
I was going to ask the same question. Is your thalamic segmentation from FreeSurfer?
It's from the Iglesias et al., which is incorporated into FreeSurfer now. So it's this kind of individualized segmentation for each person.
I asked because I am pretty sure that they also have a brainstem segmentation, but I don't remember how fine it is. I think it's pretty bulky, but even so, it might be interesting to just look in general, at what's going on in the brainstem, even if you can't.
It's been so great. Yeah, no, I would love to look at brainstem. And in fact, I don't have a plot for it, but certainly the entire brainstem is clearly showing very salient arousal lock dynamics. We're just not confident of localization yet. So there's a lot of interesting stuff in there. And I agree, it would be great to dig into it. And I think some of the nuclei probably are resolvable with the spatial resolution. Locus coeruleus is just a particularly thin one, but it's been really great. The FreeSurfer team just keeps creating new segmentations. And then happily accept them and start looking at different brain regions.
It's all really great. Thank you.
Thanks. Great. So overall, we found this kind of divergence of cortical and thalamic activity at state transitions. And within that, we found that the thalamus itself actually contains temporal sequences that unfold over about 3 seconds and actually precede the change in arousal state and behavior metrics that we can detect, which I think is both really interesting for thalamic control of sleep, but also just sort of highlights the usefulness for fast fMRI. If one is interested in looking at subsecond dynamics, it's potentially possible to actually resolve those across distinct nuclei of the thalamus and potentially other subcortical structures that are of interest, like the brainstem as well. So this is our kind of neural circuits interest in sleep. And now I'll kind of switch gears to talk about a very different thing that we measured in sleep, which is trying to understand maybe why sleep has these major metabolic consequences for the brain. And in particular, I mentioned that rodent studies had shown that waste clearance seems to change during sleep, and that levels of things like amyloid are actually declining in the brain during the night and during sleep. And so we wanted to ask whether actually potentially there's a change in cerebrospinal fluid flow that happens in the brain during sleep, because we know that the brain is kind of suspended in CSF and that this is really critical for waste removal and for the metabolic homeostasis of the brain. And so to do this, we wanted a method to actually measure CSF flow during sleep. So we wanted to both ask whether macroscopic CSF flow patterns change during sleep in the human brain, but then also, if they do, why would that be? Why is it that the state with these really distinctive neural dynamics is also the state that seems to be coupled to potentially waste removal or other kinds of metabolic processes in the brain? So this study was led by first author Nina Foltz in the lab. So this is a 3T study where we performed simultaneous EEG and fast fMRI. And so the EEG again is to let us look at sleep. And with our fast fMRI, we get the bold signal, so we can look at how hemodynamics are changing during sleep. But we also used a kind of adapted version of this to simultaneously look at CSF flow in the brain. And so we scanned 13 sleep-restricted subjects, again at midnight, using a temporal resolution of 367 milliseconds. And so what this image is showing is just this kind of special positioning of the acquisition volume that we performed, where this bottom slice indicated in yellow is really intersecting with the ventricle. And so if I blow up this bottom slice of our imaging, you can see that there's actually this really bright white signal in the bottom of the brain, which is actually indicating the upwards flow of CSF through the ventricle in this image. And so what this method is essentially exploiting is that fast fMRI carries lots of benefits for measuring faster signals, and it also carries very distinctive artifacts. And one of the classic artifacts for fast fMRI is that it's very sensitive to flow signals. And so this can be a confound, particularly if you have, for example, lots of incoming blood flow to the brain. That's obviously kind of going to be related to your bold signals in complex ways. But if you're imaging in the ventricle, you can actually potentially also convert your flow signal into a CSF flow signal. And essentially what happens is if you scan very rapidly, you keep hitting your brain with RF pulses and all of the tissue eventually reaches a steady state, but any new fluid that's flowing upwards into your acquisition volume has not yet experienced any RF pulses and will have a very bright signal because it has not yet reached that steady state. And so using this method, if you have very fast upwards flow, you'll get bright signals in your bottom few slices. If you have slow upwards flow, you'll get a bright signal perhaps just in the bottom slice. If you have downwards flow, you will not get any signal at all. So this allows us to use fast fMRI to extract a CSF flow signal that's coupled to the velocity of CSF flow in the ventricle. And so using this, we could now go in and ask whether when our participants fall asleep, is there potentially also a change in the CSF flow in the brain? So this is an example of somebody who was sleeping in a scanner. And again, this top panel is the EEG spectrogram. So for the first half, this person is stably asleep. We see these kinds of 14 hertz rhythms, which are the spindle band. Here you see this awakening, which in this case, we did throw out this data from this awakening. This kind of sharp vertical stripe in the EEG is a motion artifact as this person kind of wakes up and remembers where they are. And then they reached this stable wake period, and this 10 hertz occipital alpha rhythm kind of pops back on. That's that red stripe here. And some of these participants did our same task. So for example, this person is not doing the task. They're completely asleep. They kind of spend some time waking up here, and then they reach this stable segment of wakefulness. And if we look now at what the CSF is doing, we found that when people are asleep, there's actually these huge waves of CSF flow that are pulsing every 20 seconds or so in the brain. And as soon as the person wakes up, these waves disappear. They're replaced by this much smaller, kind of faster pulsation that's kind of constantly ongoing. This represents that CSF is actually always pulsing with every breath we take, which had been previously shown and is kind of consistently the case during wakefulness. So this is an example just from one person kind of showing this phenomenon of really large waves of fluid flow that appear selectively during sleep. If we look at the power of the CSF signal across all of our subjects, we consistently found during sleep, we had this kind of increase in flow, peaking around 0.05 hertz. So essentially these waves are really happening approximately every 20 seconds or so during sleep. So this is very interesting to us that there's actually a change in the fluid dynamics of the brain during sleep. But then the next question was, well, why? What's actually driving this kind of very large volume pulsation of CSF flow that's kind of coming upwards every 20 seconds during sleep? And so the first thing we thought to look at was the hemodynamics, because it's actually also known that when vessels pulsate, you can see fluid flow along the walls of the vessels as well. And we actually have a hemodynamic measure because we were doing bold fMRI at the same time. So here the bold signal from the gray matter is in green and the CSF is in purple. And you can see during wakefulness, there's this kind of light pulsation of the CSF. But when this person falls asleep, these huge upwards waves of CSF flow are actually very tightly anti-correlated with the bold signal. So the bold signal is also showing these huge waves of fluctuations, but when the bold signal dips, then the CSF flows upwards. And so a mechanism that would actually be very consistent with this is that there starts to be oscillations in blood volume in the brain. Our skull has fixed volume. If you have more blood, the CSF has to go somewhere. And so as blood flows into the brain, CSF would flow out. And as blood flows out of the brain, CSF would flow in. And so this is just a video of what this tends to look like in one example participant who's sleeping in the scanner. And in this, the red will be the bold signal, and the blue, which is measured just here in the ventricle, will be the CSF flow. And so what you see when someone's sleeping is you have these kind of characteristic waves where the whole cortex is showing this bold wave. And at the end of each bold wave in red, then you see this upwards pulse of flow in blue of the CSF. So first you have the bold, and then you have the CSF flow as it ends. And then you have the bold, and then it's followed by the CSF flow. So these patterns seem to be very coupled. We have these kinds of really large fluctuations in bold, followed by this upwards flow of CSF every 20 seconds or so. So we went back to ask what might be actually inducing these huge bold fluctuations. And of course, as neuroimagers, we're used to actually thinking about the source of bold as being the neuronal activity of the brain. And so we looked at whether something might actually be changing in the EEG when people were having these huge bold fluctuations. And so what this plot is showing is the time course locked to the peak of each of these CSF flow waves. So you can see time 0 here is when you have this really strong upward CSF flow. It's coupled to this kind of large dip in the bold signal that we see throughout the cortex. But about sort of 5 to 6 seconds earlier, we actually saw this really striking increase in the slow delta power of the EEG. And these slow delta waves of the EEG are actually the defining feature of sleep, is the appearance of these really low frequency rhythms in neural activity. And so this could actually, again, be really consistent with this idea of neurovascular dynamics being linked to fluid flow. We know from prior studies that if you have a slow wave during sleep, it represents that neuronal activity in much of the cortex becomes silenced at the same time. When you have an EEG slow wave, many neurons in the cortex are all simultaneously suppressing their activity for hundreds of milliseconds. And so it would perhaps be unsurprising that after a slow wave in the EEG, you would get this kind of really substantial dip in the bold signal throughout the cortex. And then after that, you then in turn see this upwards flow of CSF, which again would be consistent with this idea that when you have a dip in blood volume, the CSF will actually kind of flow inwards to compensate. And so based on this idea, we've asked could we actually deconvolve a relationship that takes us straight from the neural signals we're measuring in the EEG to predict the signal that we'll actually see in the CSF flow. And when we deconvolved this impulse response between the EEG and the CSF flow, we got this very kind of beautiful characteristic function, which looks a lot like a hemodynamic response function. So after EEG slow waves, the best fit prediction of subsequent CSF flow was modeled by this kind of slow increase, which peaks about 6 seconds or so after we see the EEG slow waves. And again, this is just sort of a computational model-based method. There's many things that change when people have slow waves during sleep, but it seems like at least part of the mechanism could be that the neurons are actually changing fluid flow in the brain due to their effects on vascular volume. So to wrap up this last part, we found that there's very large waves of CSF flow that appear during non-REM sleep in humans. Most of our data is from N2 sleep. It's a sort of intermediate sleep stage that we spend most of our time in. We haven't imaged REM sleep, so we don't know yet what's happening there. I think that's going to be really interesting to look at in the future. And the timing that we see actually suggests that neural activity could potentially drive CSF flow because of its consequences on the vasculature. That neurovascular coupling gives neurons a way to actually also directly impact the flow of CSF throughout the brain. And we were very inspired to look at this because of these studies on waste clearance, but I think it's also really important to note that we don't actually measure waste clearance in this study. And so we still need future studies to actually say, okay, well, if you get this very large scale CSF flow, what does that mean for the metabolites in the tissue and in the fluid that could potentially be being transported or not. And I think computational models will be really useful for that as well. All right, so to conclude, We would argue that multimodal imaging at high spatiotemporal resolution can really reveal new aspects of information about brain dynamics, both measuring faster timescale information about what the neurons are doing, as well as letting us uncover relationships between neurons and other kind of physiological systems of the brain, like fluid flow. We found that the transition to awakening at sleep, at the end of sleep, is preceded by a sequence of activity and a specific constellation of thalamic nuclei, kind of uncovering temporal sequences that precede these changes in behavior that are really striking across brain states. And we also found that neural activity and CSF flow are coupled during sleep, and both are kind of having large slow waves together, which suggests potential mechanistic pathways that could regulate CSF flow in the brain. And I'd like to acknowledge my fantastic lab members at BU, especially Nina Fultz and Beverly Setzer, whose work was in the slides that I showed you today, as well as our great collaborators at the Martino Center at MGH and our funding support. And I would love to take any other questions.
That's great. Thank you so much. All right, so let's start by questions from students, if there are any.
Hi, Laura, I have a question for you. I have many questions actually, but thank you. This was a fantastic talk. I loved it. But I had a question about the thalamic sequence when you see people waking up. Have you looked to see after you see that peak and it drops back down, if there's a sort of a reverse sequence or what happens to those thalamic nuclei on the other side of that peak, or if it differs between sustained and transient sleepers or wakers?
Yes, great question. So we haven't tried to look at the other side because there's not always enough time to separate like what would be the awakening and whatnot, but we have looked at the counterpart, which is sleep onset. And actually it's much harder to define a time of sleep onset because it's much more gradual. But if you take the same just behavioral approach, you look at the last button press, you actually can also see signs of a sequence there as well, but it doesn't seem like it's the same. It shares some properties, but not all. It's like some of the same nuclei are involved and some are not. And so we're still working on that. We did also look at the comparison between people who have sustained arousals and people who have these transient ones where they just wake up for a moment and then they fall back asleep. And that actually also seems to differ in a sort of interesting way. It seems like when people have the sustained arousal where they really manage to stay awake, the sequence actually happens much faster. It's as if there's a more kind of effective rallying of these nuclei and they're activating in a more sort of unified and coordinated way. Whereas in the transient arousals, it seems to be this much more kind of sluggish sequence, and then they end up actually just lapsing down again.
That's fascinating. Thank you.
Yeah, fascinating work. Just a quick question for you. Have you looked at the correspondences between the CSF and only the slow wave sleeps, or did you try and look at whether or not they map onto, say, alpha waves when it's like in light sleep or stuff like that?
Yeah, that's a great question. So we've only subdivided like the very lightest stage of sleep, N1 and N2. We see that there's more CSF flow in N2, but we haven't tried to kind of disassemble how different EEG rhythms might be differently related to CSF flow. And I think you're right. Like, I think that probably that relationship is state dependent. It's also tricky because actually these EEG rhythms are correlated with each other. Like when you see a slow wave, you tend to also see a spindle, you tend to also see other kind of phase locked rhythms. And so I predict that you would actually probably find multiple brain rhythms that are correlated with this. And then in terms of trying to figure out whether one is the key driver, my prediction would be that essentially anything that can drive very coherent changes in neuronal activity, anything that could drive a hemodynamic response effectively could potentially drive this flow effect. So I can't see a reason why it would actually need to be a specific frequency other than the fact that the slowest ones tend to be the ones with the largest changes in neural activity and that are the most effective at driving neurovascular coupling.
Thanks.
Okay, other questions?
Hi, Laura. I have a question. I hope it's a little tangential, but hopefully not too much. I'm curious about what happens when people wake up. So, this idea that sleeping is sort of cleaning things out is really interesting. But when people wake up after a night's sleep, they're often quite sluggish for a while. What do you think is the main mechanism that causes that delay in getting things up to sort of optimal running order again?
That's such a good question. I wish I could answer that. I really don't know. This is one of the challenges we have during sleep, right? Is how do you actually get a sort of quantitative measure to represent it? Like, I would love to know the answer to that if there was a way to disambiguate when somebody's awake and sluggish and when they're not. I think probably you would need to use a stimulus, which is really tough in sleep because you can accidentally wake somebody up when you didn't mean to. One possibility is that there's a kind of fast change in thalamocortical dynamics with the kind of switch back to the awake desynchronized rhythms, but not a fast change in like the neuromodulatory state, right? All of the kinds of different levels of noradrenaline and, these other kind of neuromodulatory and acetylcholine and these others may be slower to adapt, but that's a very fuzzy answer. I think basically, I don't really know. I think it's a really interesting question. Yeah. It would be cool to design just the right experiment to try to get at that.
I have one more question. So Laura, first of all, great presentation. I think, there are questions that you can answer, but the questions that you did answer were done so thoughtfully and well controlled. I really enjoyed the presentation. So thank you for that. My question is, as someone who's doing and incorporating EEG with fast MRI, and that's something that we've been wanting to implement at our center, what are some of the challenges that you faced during this work that you've done?
Yes, that is actually surprisingly challenging. So when this first started, none of the EEG companies would allow you to do fast fMRI with well, no, they wouldn't not allow you. None of them would validate that you can do EEG with fast fMRI. So the concern always when you do EEG inside the MRI scanner is that you're introducing the possibility of heating. Because you have metal, you have kind of conductive loops being formed in the scanner. And so because of that, initially, they basically said you can only use this with kind of standard old school bold. But if you want to do anything else, it's on you. And so I was working with a collaborator at the Martino Center, and we basically did a bunch of in-house kind of measurements on agar phantoms where we could insert lots of thermal probes at all the different locations and assure to ourselves that in fact, there's not a heating effect from using this. And so basically every site does need to kind of determine for themselves their own safety performance with their own equipment and their own kind of local situation. That said, a few years ago, I think the EEG companies realized that actually this is okay. And so they no longer prohibit the accelerated multiband sequences. Instead, they set the limit based on the actual power deposition level of an individual sequence. And so in fact, there's plenty of very accelerated sequences that are pretty low power, pretty low SAR. So now we have much less challenges because, our fast imaging actually does happen to fall within the recommended range for the EEG company. But you always want to kind of validate the safety question before embarking on an EEG and fast fMRI study to make sure that it's within guidelines, that it won't cause issues.
Right. Yeah.
Yeah, it was a surprising complication that we had not anticipated. It took a lot of gel phantoms to resolve.
Does anyone else have any other questions? All right, well, if you do, please feel free to email Laura and join me in thanking her for this wonderful, wonderful talk. Thanks, Laura.
Thank you so much for coming and for your great questions. I really enjoyed meeting everyone.